Okay, students, as we continue to talk about microorganisms in the soil, here we're going to talk today about the algae and also the viruses, as they can be within the soil environment as well. The algae, they're are well known for their ability to harness energy from sunlight and then transfer inorganic carbon dioxide into organic form like glucose, which is a process known as the photosynthesis. So they are photoautotrophic organisms. They can vary greatly in morphology, so they can be differentiated from one species to another by their shapes. Also, we can divide them according to the physiology, reproduction, and habitat. The general characteristics for algae is that first they're photosynthetic, they evolve oxygen and they can fix carbon dioxide. In soil, most crucial is needed is water and light. So having some moisture and light, there's a good environment for algae to grow. Their shape is usually unicellular or filamentous. The cell wall is diverse. Cell wall could contain cellulose. Other genera may have silica on their cell wall. The algae could span a wide temperature range, environments where they can grow from sub-zero to around boiling temperatures. In the environment where the terrestrial environment, we could see the activity of algae usually on the surface of the soil, because in the subsurface, they're usually dormant, or they could acquire their energy from facultative organotropes, from organic chemical transfer. But most of their establishments are usually on the surface of the soil. We can classify algae according to the two major groups. One is defined as the eukaryotic algae, which are part of the plant kingdom, and the other one is the prokaryotic algae, which is, for example, we have the cyanobacteria, is part of the prokaryotic algae that could have prokaryotic cellular organization. The cyanobacteria were previously known as the blue-green algae. Now they're giving the name cyanobacteria, and they could have characteristics close to prokaryotic bacterial cells. The major groups in soil, we could start with the cyanobacteria and their major ability to fix nitrogen. The green algae, they could be filamentous in shape. And another group that we mentioned that they have the silica cell wall, they're called the diatoms. And we're going to see some of their shapes as well. And the yellow-green algae. The primary algal divisions, they said the green algae and the diatoms. You can note here, this is the volvox, where they could colonize together the cells, or they could be filaments. They're usually photosynthetic, and they're usually unicellular. The other group, they are the diatoms here. They have the silica cell wall. They're usually symmetrical. Like if you have to divide one cell in half, you could see that this half is similar to this half. So there's symmetry in those cells. The picture here on the bottom represents the diatoms, where cells are usually symmetrical due to the silica cell wall arrangement. So algae can be divided in the soil according to their pigmentation, which is the color of the pigment. The pigment is there to capture light from sunlight, to capture the energy from sunlight and use it in photosynthesis. We can divide algae according to their cell wall composition, whether they only have cellulose or whether they have silica within their cell wall. We can look at their cellular organization, whether they could be prokaryotic or eukaryotic algae. We could also look at the storage products. what form of storage organelles or granules that they use to store extra food that they can synthesize. Also, we could look at the flagellation, the type of flagella, and the number of the flagella. The presence of algae in the soil is important in the terrestrial environment. Usually, algae, they can form symbiotic association with fungi in an organization called the lichens. And those lichens, they're often called the primary colonizers. Other than being the primary producers, they produce food through the photosynthesis. As the lichens grow on rock surfaces or on some ruins and artifacts, fungal hyphae can secrete extracellular enzymes that can have another characteristics of biological weathering to acquire minerals like phosphorus and other nutrients needed for the algae. And if we look at the cyanobacteria and its ability to fix nitrogen, this is very essential for the soil, since nitrogen is the limiting growth factor. And by adding cyanobacteria to soil, as if we're adding biofertilizers, and it's a good industry in that regard. The algae is well known for the stabilization of the soil aggregates through the secondary metabolites, through the polysaccharides or glycoproteins. They can fix nitrogen in photosynthesis by transforming CO2, inorganic carbon, to glucose as in organic carbon. And algae, they are indicators of environmental quality. The picture here that you see, there's a wide growth of green on the surface of this water body. This indicates pollution, extra nutrient load with nitrogen and phosphorus. So as eutrophication exists, it's an indication of a bad or poor environmental quality. On the surface, the algae, they can form what is called desert crust on the surface that prevents erosion. But as the moisture becomes too little, the algae became dormant most of the time. The relative number and biomass between bacteria, fungi, and algae, you could see here that algae are very less dominant in the soil. Their numbers greatly decrease as we go through the profile, and they're also, the biomass does not contribute much to the biological mass in the soil. But their requirements and environmental effects such as moisture, light, acidity is also required for algae. Most diverse community of algae, they occur between 7 and 8. For example, like diatoms, they prefer a pH range between 7 and 8. They also need nutrients such as nitrogen and phosphorus. As for carbon, they can fix their own carbon through photosynthesis. The effect of other pesticides, they're very sensitive to herbicides, for example, because they could uncouple the photosynthetic system. They could be an indicator, in that regard, indicators of environmental contamination. Okay, that's for algae. As for viruses, the virus, they are obligate parasites. They contain one type of nucleic acid, either DNA or RNA. The viruses, they have specific host. So not one virus could only infect or a single type of cells. They have a limited host range. Most plant viruses, they have RNAs. Those viruses only infect plants. And for the viruses that infect bacteria, they're called bacteriophages. So a bacteriophage is a virus that can infect bacterial cells. inside the cell, the virus is biologically inert. It relies on host for replication and to synthesize cellular requirements for the virus by using the host to copy the RNA of the virus. So the viruses are the smallest. They can't be seen by compound microscope. Therefore, we need the more sophisticated technology like electron microscope in order to see various shapes. As you see here, different types of viruses have different shapes, and those images can be taken by electron microscopy. Their significance in the soil: they're important for gene transfer. They can regulate microbial population. As the bacterial cells grow, they don't grow, population does not grow forever. Well, there's a natural resistance to how far a population can grow. With the presence of viruses in the soil, with a proper host bacteria, if they both are there, then they can decrease the bacterial population. Some viruses are still disease and public health concerns, but in the soil somehow they act as a purification system. The soil by itself inhibit or inactivate the virus activity, okay, inside the soil, because they don't always match between the virus and its host. That's what we have for the viruses, and thank you for listening.